The In-Nucleus high-C protocols allows the exploration of chromosomal conformation at different resolution for the characterization of chromatin loops, domains, and compartments organizing the genome.
This protocol provides high-resolution profiling of genomic interactions at different scales, yielding a more consistent coverage over the full range of genomic distances and data with less technical noise.
Combining this protocol with genetic editing is a powerful strategy for testing the structural function of genetic elements. It can also be applied to the characterization of structural variations that lead to disease. The In-Nucleus high-C protocol can be applied to explore the genome organization of any eukaryotic genome.
During SDS and Triton treatment, disaggregate any nuclear clumps by pipetting, as aggregates interfere with enzymatic reaction efficiency, and be sure to perform the appropriate quality controls measure prior to sequencing.
Begin by adding methanol-free formaldehyde to 1 × 10⁷ Schneider's line 2 plus Drosophila cells in 17.5 milliliters of Schneider medium supplemented with 10% FBS to a final concentration of 2%. Incubate the cells for 10 minutes at room temperature with mixing every 60 seconds or on a rotating wheel, and add glycine to a final concentration of 0.125 molar with mixing. After 5 minutes at room temperature and 15 minutes on ice, collect the cells by centrifugation, and carefully resuspend the pellet in 25 milliliters of cold PBS, and collect the cells with another centrifugation.